97, metabolic activity is just in front of lab 15, which is the first lab we're doing today. This sort of gives you the background for what we're doing in the rest of the semester. So the first half, you became familiar with what microbes look like under the microscope. You learned aseptic technique, how to inoculate, how to get isolated colonies. That's basic lab procedure. From now on, we're going to focus not so much on procedure. You've got all that down. We're going to focus instead on identifying bacteria. Right? So if you're working in microbiology in real life, you're in a hospital lab, a health department lab, something like that, where you've got a sick patient who maybe has diarrhea or some other condition, and you're trying to figure out what's making them sick. Right? So you get a sample from the patient. It's always a mixed culture. Whether you're getting a sample swab from the back of somebody's throat who the doctor suspects has strep, or you're getting a fecal sample to figure out what the problem is with diarrhea, there's always a mixed culture of lots of different kinds of bacteria in it. Some of them are supposed to be there, right? We've got bacteria that are supposed to be in your intestines and aren't making you sick. So the first thing we've got to do is do that streak plate, separate them out, and then figure out which one of those is the one causing the problem. There's no real easy way to do this. In this day and age, DNA technology is coming on board, making it a little bit easier, but the equipment you need to identify bacteria based on their DNA profile is pretty sophisticated and expensive. So there aren't many labs that do it that way. So you're going to learn what most labs do. We do a metabolic profile. We find the bacteria from that mixed culture that we think might be the one causing the problem, and then we figure out what kind of food does it eat, what kind of waste does it make, what kind of byproducts does it make, and by looking at that whole profile of its food and its waste, we narrow down which bacteria it is. Okay? So you're going to have an unknown sample, probably from a patient in real life. Not going to have patient samples in here, obviously, but at some point in the semester you'll have an unknown bacteria, and you'll have to figure out which one it is. So before you can get there, you've got to learn what the tests are that are done, the metabolic tests, which is what we're doing on page 97, and if you flip over to page 98 as well. So the first set of tests that we're going to be doing are polymer hydrolysis tests. This is the next 4 or 5 labs. Luckily, you already know what this is from lecture, right? What's a polymer?
A chain of
many subunits, right, as opposed to monomer. So in lecture, you talk about 4 major macromolecules, right? Carbohydrates, DNA, protein, and lipids. Those are your polymers, right? Large molecules. And what was hydrolysis?
water.
Adding water to do what to them? Break them down, right? Add water to break the bond. Now, a little bit different spin on it here in lab. When we talked about it in lecture, we didn't really focus on this. Bacteria are so small that they can't do hydrolysis internally, right? You gotta have to climb 1,000 times to see them. They can't eat or engulf a big piece of fat, a big piece of starch, and then digest it internally like we would, right? We chew it and swallow it, and our stomach does the digestion. They're just too small. So what they're going to do instead is secrete enzymes to the outside of their cells to do this hydrolysis externally, break it into small pieces outside, and then they absorb those small pieces and use them for food. Okay? So this is going to be external for bacteria. And those enzymes that they secrete are called exoenzymes because they're being secreted, they're exiting the cell. Enzymes secreted to the outside of the cell in order to do polymer hydrolysis. Okay. Enzymes secreted to the outside of the cell in order to do polymer hydrolysis. Because again, they're just too small to do it internally. So it's all external. So again, the next 4 or 5 labs are going to be polymer hydrolysis labs, and the major difference between them is which polymer you're using. Okay, so in lab 15, if you're over on page 99, lab 15, we're doing starch hydrolysis. So what's our polymer here? Starch, right? That's the big molecule, right? The exoenzyme that breaks it down is called amylase. actually the same enzyme that we use as humans to break down starch. Amylase is in our saliva. Bacteria make it too, except again, they're secreting it to the outsides of their cells. Now, not every bacteria can make amylase. Some bacteria can produce it, some can't. That's the point of this test. We're going to be looking at the bacteria listed on page 100 in your material section. We've got Bacillus subtilis and E. coli.
What page?
At 100. Page 100 material section. So we're going to be checking those two bacteria to find out whether or not they produce amylase. That's the point of this test. Do the bacteria produce amylase or not? Because again, in real life, you've got an unknown patient sample. We're just trying to get an idea of what they eat, what waste products they make, and this is just the first test we're going to do. So the way we're going to find out, do they make amylase or not, is we're going to put them on a petri dish that contains starch agar. It's basically just nutrient agar that we've added some starch to, like cornstarch or potato starch was mixed in with the agar. What you should do first is get one of those Sharpie markers and divide the plate up into 3 sections. Use sort of the peace sign to give yourself 3 sections. Make sure you're labeling, you're doing this on the agar side. Remember, you never want to label on the lid, the side that has the agar in it. One of those 3 sections is going to be your control, that you don't put any bacteria in, right? The other 2 sections are for the 2 bacteria that are listed. So you've got, I think, B. subtilis, is that the one?
E. coli on that side.
Yeah, well, it doesn't matter. E. coli, what's the other one?
B.
It is subtilis, okay. So you label which bacteria you're putting in which section. And again, remember, try and write small off toward the edge. Right? You don't want to block your view. We're not streaking this plate, though. We're not trying to get isolated colonies. Okay? You're going to use your aseptic technique, flame your lube, flame the openings of your stock culture there on the front table. This time your stock cultures are on slants. They're not broths, so you're scraping up the bacteria that are growing on the slant. And then you're just going to draw one small straight line in the Bacillus subtilis section. Again, we're not trying to get isolated colonies.
That petri dish already have agar
Yes,
in it.
and it's already solid.
Okay.
Yep.
Okay. Picture.
Yep, no problem. Again, flame your lube, aseptic technique, put one small straight line of E. coli in the other section. And that's all you're doing today. Easy peasy. You're just inoculating them. And you're going to put them in the incubator. What temperature?
37 degrees.
Yeah. And how long?
15 to 20 minutes.
Nope, that's autoclave. 18 to 24 hours. Good. And while they're in there, they're going to grow, right? They're going to go through binary fission. So when we come back next week, you're going to be able to see that line where Bacillus is and the line where E. coli is. But let's assume for a second, we're just doing this hypothetically. Let's pretend that E. coli is the positive. I don't honestly remember which one's positive. Okay, so let's pretend it's E. coli. When they're in the incubator, if there are positive bacteria, they're producing amylase, right? They're secreting it to the outside of the cell. And because agar is kind of porous, it's going to move away by diffusion, right? They're releasing amylase, it's going to diffuse through the agar. So all of the starch around that bacteria should be gone, right? Amylase should have done hydrolysis, broken it down into sugars. We're not going to be able to see that when we come back on Monday. Because the starch agar plate's the tan one over there. It looks just like nutrient agar. It's going to look just like that when we come back on Monday. So it's not going to be easy for us to tell whether or not they've secreted amylase. So to make it easier, after incubation, we're going to add a reagent. Okay, just that word reagent by itself, it's a chemical that we're adding to help us determine positive from negative. Okay, so a reagent's going to help us determine, chemical that's going to help us determine positive from negative results. The specific reagent we need for this test is iodine.
Then the iodine transfers through. Oh no.
It does.
Oh, I remember that.
Well, that's what we're
The reason that the positive or negative result, that's telling us if they're sick or not, right?
Well, no. We haven't even gotten to that point. If you're really doing this in the real world, they're already sick by the time you get this sample. They're definitely sick. We're trying to figure out what they're sick with.
Okay.
Yeah. gonna go around swabbing
So
healthy people.
what's the positive or negative for? I'm just trying
Well, I haven't told you yet.
Oh, okay.
Hang on. No, you're looking at your learning objectives.
No, I don't even have.
Okay. We're not there yet. Yeah, so far we've plated the bacteria, we put them in the incubator to grow. After 18 to 24 hours, we're going to add the reagent iodine. You're going to wait till Monday so you can see it, right? We'll put them in the fridge for you. Iodine, like Jamel said, when you mix iodine with starch, it turns blue-black. What color is iodine normally?
Red.
Well, it's sort of an
Brown.
amber, sort of a
Yeah.
brownish yellow, kind of amber color. But when you mix it with starch, it turns a really dark blue-black. And in our labs, it's always been more toward black than blue. So this control section of the plate, you didn't put any bacteria in. It's just starch agar. So when you add iodine, what should happen to this section of the plate? It should turn black, right?
What do you
And
mean,
if
black
we're-
light?
Like
What do you
the
mean?
color of these tables, the whole thing's gonna
Because
turn.
nothing's there
Right,
but the agar. Okay.
right, there's no bacteria in there, it's just starch agar. So this bacteria, in this hypothetical example, didn't produce amylase. So should there still be starch around them? Yes. So what color is that gonna turn? Black. You're gonna have black color all the way in that section of the plate. Because that bacteria didn't produce any amylase, the starch wasn't destroyed. But these bacteria produced amylase. There's no starch inside my oval there, right? Amylase has destroyed it all. So is it gonna turn black?
No.
No. Okay, so you're gonna have what looks like a halo almost around the bacteria that isn't black. So, our positive results, what positive looks like, no black color around the bacterial growth. Don't just write halo. I need to know that you know what color it would have been. Okay, so positive is no black color around the bacterial growth.
No black color.
No black color around the bacterial growth. No black color around the bacterial growth. What does that tell us about the bacteria? What does that mean?
That hydrolysis happened.
They produced what?
Amylase.
Amylase. So positive looks like no black color around bacterial growth. Positive means that they produced amylase. So what's the negative result look like?
Black.
Black.
It has black color around the bacterial growth. Okay. And that means
No amylase.
it does not produce amylase. Okay. So things to study for next Wednesday's quiz, right? Because they're still on Wednesday, so next Wednesday. You want to know, obviously, the name of the test and the reason you're doing it. Find out if they produce amylase and can do starch hydrolysis. The name of the exoenzyme, right, which is amylase. The name of the media that we use, starch agar, for this one. Any reagents, this one's got the reagent of iodine, right, to help us tell positive from negative. What positive and negative look like. Positive, no black color, negative, has black color. And what positive and negative mean, what they tell us about the bacteria. Positive tells us they produced amylase, right? Negative tells us they didn't. And that's all in your learning objectives. Look at that whole list of things you need to know. Now, if you've got that photographic atlas, right, bless you, that you were supposed to purchase at the beginning of the semester, the starch hydrolysis test is on page 92. If you don't have it with you, just write it down. Page 92 is where it is, so that you can read through some of the background, and there are a couple of pictures there showing you a bit like what this looks like. Some questions on that? It takes me longer to explain it than it's going to do for you to set it up today. All you're doing is making 2 lines of bacteria. All right, so we're doing one more lab. Since that one's nice and short, we're going to